Good evening or morning whenever you're watching this. This is a lecture being recorded for JS 110 Introduction to Forensic Sciences at UAB. This is the lecture on forensic DNA analysis, part two of a two-part lecture. My name is Jason Linville, and this is being recorded, the last bit of information. In the fall, we just established it's September and everybody's sick. Fall of 2017. So if you're accessing this archived lecture in future semesters, all the material still applies. But if I mention anything about being sick or hurricanes or the NFL or the president or Dwayne Wade playing and not being injured for Cleveland, then you can ignore all that information and just focus on the material. Now, the good thing about part two of the two-part series, for those of you attending live tonight, is that we've already gone over the what is in part one. It's what is DNA, where is it found in the body, and how does it differ among individuals. So now we actually start on what happens in the laboratory. So we don't have to go over a case. But just to review for everybody, if we were to go over a case, it would be where we're comparing the traits of DNA, which, as I may have hinted at in part one, is going to be 13 pairs of numbers. And we are going to get 13 transfer a blood evidence from the crime scene into 13 pairs of numbers. And then we are going to transfer our suspect's DNA into 13 pairs of numbers. And then we're going to see if they match. So what we want to go over today is just a little bit about the process in the laboratory, but we're not going to go into a lot of detail. In the class JS 352, one of the forensic lab classes, that's where we go into a lot more detail about the DNA analysis and, blood and body fluid testing as well. But for here, we're just going to give you a general idea. So hopefully you can understand, again, it's all about understanding where those 13 pairs of numbers are coming from, and also why we can say they're specific to an individual, even though they might not be identical twins. Okay, so the unknown samples that come into the lab can be a variety of things. I mean, obviously, blood stains, semen stains, whether the semen is on bedding or clothing, or whether it's on a swab from a sexual assault kit. Saliva residue. This would include, if you're sampling saliva from a plastic cup or cell phone or anything else. And all these other materials, hair, tissue, bones, teeth, sweat, as we covered in part one, it's anything that contains cells. If it contains cells, it contains DNA. And if you're targeting those other types of samples, you need to target where the cells are. So those unknown samples we're going to transfer to 13 pairs of numbers based on the DNA that's inside of them. And then we get a known sample from our suspect. It can be blood or buccal swab. Normally it's a buccal swab. What is a buccal swab? It's just a cheek swab. So it's just you take a swab, collect cheek cells from your suspect, and then you process it and turn it into, again, 13 pairs of numbers that represent the DNA. If you are convicted of certain crimes, or in Alabama, if you're arrested for certain crimes, the lab will take a swab of your cheek cells and, again, process that, do DNA analysis, convert it to 13 pairs of numbers, which it then stores in a database. So in the future, if your body fluids turn up at a crime scene, you can compare them to a database as well and get a match. Okay. When it comes to analyzing DNA, you have to be very careful that the investigator or the lab analyst doesn't contaminate the DNA, or that you get one sample mixed in with another sample, and that would also be considered contamination. Anything that happens after the sample is collected that involves getting two types of DNA mixed together would be referred to as contamination. So I think we went over this a little bit in the crime scene investigation, but it's worth re-mentioning here that you want to sort of protect the evidence from your own DNA. So obviously wearing gloves, using disposable instruments will help prevent cross-contamination. Not sneezing on a sample, but sneezing away from a sample is a good idea, or sneeze into your sleeve, again, with the illness season coming up. And Also, the worst type of contamination that can be made from the view of the forensic scientist is if you get an innocent person's DNA, a suspect's DNA, mixed in with the evidence, because then it places that person at the crime scene when they weren't actually there, and it can lead to the conviction of a guilty person, which is, again, what forensic scientists want to avoid at all costs. So in order to do that, the known samples, in other words, if I'm your suspect and you have blood at a crime scene, you're not going to take my cheek cells, and you're not going to put it and store it alongside the evidence. You're going to keep them separate. In fact, if it's possible, what you'll do is the lab will process the evidence first before they even take my DNA, my cells, out of the evidence locker. That way, there's not a risk of them, processing my DNA and accidentally contaminating a pipette or getting it into another tube. It's where they get the profile from the evidence first and then get it from the suspect. All right, so you want to package these, and again, this all involves prevention of contamination, as well as prevention of the evidence being destroyed or the DNA being destroyed or degraded. Each item you want to keep separately. Paper bags, as we said before, with the evidence packing, not plastic bags. Paper bags allow the biological evidence to dry out, and once it dries out, bacteria cannot grow, and therefore the DNA is protected. Okay, here this is sort of skips some steps here. Okay, it's the moisture being trapped that will lead to the degradation of DNA, but actually in between here, the moisture allows bacteria to grow, and it's actually that bacteria that destroys the DNA. Oh, that's a terrible way of writing bacteria. All right, so you want to keep samples dried out. Once they're dried out, they can be held at room temperature. They can be held in a refrigerator, which will prevent further degradation. So anything that you can do just to prevent degradation, cool, dry place is what we're looking for. Okay, so at this point, let's imagine that a blood stain is in the evidence locker. A buccal swab is in the evidence locker. A sexual assault kit is in the evidence locker. We want to go and let's say it's already been processed with the presumptive test, so we know it's blood. We know there's semen present. And usually after those presumptive tests, the evidence goes back in the evidence locker until it's time to analyze the DNA. Okay, so we're going to analyze the DNA in the lab. What are we actually going to do to that evidence? Well, to put the current methods in the proper perspective, I guess, I don't quite know how to phrase this, but I do want to go back and look at a little bit of the history of DNA analysis. Now, oftentimes this is very boring because especially the two methods on this slide are not done anymore, but for anyone going into the criminal justice field, and this may even apply more to criminal justice majors who are possibly looking at becoming attorneys or going into the court system than the forensic scientists themselves, it's important to understand the history, because you'll know when DNA analysis was done on samples and when when you can go far enough back to where it wasn't the common way of doing things. Specifically, what I'm really talking about is the DNA Innocence Project, and having cases that have been appealed because they want the defendants or the convicted person wants to go back and reanalyze evidence from their case using modern technology. Okay, so you may have heard this. Reanalysis of evidence sets man free after 20 years in prison. Okay, the time frame on all of this is before 1985. DNA analysis was not demonstrated, it was not performed, it was not really known of that it could be done. I mean, sure, there were a couple theories, but 1985 was really the first demonstration that you could analyze DNA, and from evidence you could get a profile, and at that time it was called a DNA fingerprint, which we'll figure out why that is, and you could link it to an individual. Okay, not just blood testing, not serology or blood proteins or anything like that, but actually using the DNA to link evidence to an individual. So prior to 1985, you may have still collected biological evidence, because you still may have done blood typing that we talked about a couple lectures ago, or some other type of serology to help narrow down the number of suspects. But nothing was as specific as DNA, which again started in 1985. The problem is, this was more a demonstration that a paper in Great Britain was published. And what the paper actually showed, it's this first image here was the 1985 image, is they showed that, and this is a somewhat complicated thing, so let's narrow down all the non-matching parts. We want HINF1, we want the number 1, and we want probe B down here. So we're actually looking at that second column there. And it should match HINF1, number 1 here, and probe B down here. So it should match that first column. And if you compare the second column here to the first column here, and I'm aware this picture is a little small, you can sort of see how, yeah, there's a band, and then if you go down a little bit more, there's 2 bands, and then a little bit more, there's a thick band. And you'll notice that the pattern in those 2 columns matches. It's not a perfect match, but there's definitely the spacing and the number of bands there match. And this was done by chopping up the DNA from the evidence, or the DNA from the suspect, if you were comparing to it, chopping it up with a restriction enzyme, so chemically chopping up, not physically with your hands or vortex or anything like, or blender. So chopping up with a restriction enzyme chemically, and then smearing it out on a gel, which this image represents. And in reality, the DNA was chopped up into a bunch of different sizes, and you had a huge smear of DNA going from the bottom all the way to the top. All right. But the problem is that smear, when it was on a gel, DNA, you can't see DNA. So instead, what they would do is they would use a probe that bound to repetitive DNA, although that's not that important, and the probe would light up certain areas of DNA. So whenever you took my DNA, whether it was from a blood stain or DNA from my mouth or DNA from some other body fluids, if you extracted that DNA, smeared it out, and visualized it with a probe, it would always produce the same pattern. Hence the term DNA fingerprint, and it was specific to me. The problem was they thought it was specific to an individual, but they really didn't have a lot of statistics to back it up, and they weren't sure if, well, what if 2 people sort of have a similar pattern? But it didn't really matter, because by the time the late 80s and early 90s rolled around, they actually simplified the process by using this type of DNA analysis here. It was still where they would chop up the DNA. It would still be where they smeared it out on a gel, but the probe they used was a little bit different, where it would just visualize one area of DNA. And that area of DNA, since everybody has 2 copies of DNA in their genome, do we remember that from part 1? It would have 2 bands. One band came from your mother and one band came from your father. And again, every time you smeared out my DNA and visualized it with this probe, 2 bands would form, and they would always be in the exact same place. Okay, now the problem with this is that 2 people may have the band show up in the same place, but if you then used a different probe, you would get a different gel image with bands in 2 different locations. Okay, and then if you used a different probe, you would get 2 bands in a different location, and you could actually, if you ran enough of these gels and probes, you could get a combined pattern for an individual that was very specific to that individual. All right. The big problem with this type of early DNA analysis is that you took the DNA that was present in the sample, you chopped it up, but you weren't amplifying it. So if you had a very small sample, degraded sample, the bands on this gel may be so faint that you couldn't see them, and therefore the analysis was not possible. Okay, so it wasn't very sensitive. In the mid-90s, just when really labs in the U.S. never did this first multi-locus RFLP that was began in 1985, because DNA analysis had never really caught on as far as, hey, this is something that all labs should do. But laboratories, forensic science laboratories, started to do this type of analysis in the early 90s. Not in every state, but they did it in Alabama, and many other states started building databases and gel images based on this technology. Okay. Also in the mid 90s, what happened is, and actually coincidentally it was the year 1985 that the polymerase chain reaction was invented, but in the early 90s, mid 90s, forensic scientists began to use a different method that involved amplifying DNA. Okay, and this was good because it was much easier to take a small sample, an evidence sample, and amplify the DNA in it and get a profile. So where the previous gels may fail and have been too faint to see anything, now you could amplify the DNA and you could actually have a very good chance of visualizing it using this probe card method, for lack of a better term. So it wasn't officially, it was DQ alpha probe, I think is what the test was called, but it was not on a gel. It was sort of like this strip of paper, all right? And this is similar to the antigen antibody reactions we've talked about. You would wash the DNA onto this paper and then visualize it with a probe. But the important thing to understand about this test are several things. Okay, number one, mid-90s. Number two, what happened in the mid-90s? 1994, O.J. Simpson happened. And it's important to understand historically that in the O.J. Simpson case, they weren't using the same type of DNA analysis that they would use if that case happened 4 years later in 1998. All right. So they were using this DQ alpha test, was one of the tests they did. I think they attempted some of the RFLP tests that we had in the previous slide, but they did not work very well. I can't remember the exact details of the case. But this test worked very good because it amplified DNA. The problem is every person had a profile, like your profile and my profile would be where our profile would consist of two numbers, and those numbers would either be 1.1, 1.2, 1.3, 2, 3, or 4. So you only had 6 options, and you had 2 of those numbers. So what does that mean? It means it wasn't unique to an individual. If my profile, let's say that I was this first strip here, and you don't need to know how to read it, and this is also why it may have been a little bit confusing to the jury when some nerd gets on the stand and goes, okay, everybody has 2 numbers, and those numbers are either 1.1, 1.2, 1.3, 2, 3, or 4. And some jury member raises his hand and says, why isn't it just 1, 2, 3, 4, 5, or 6? That's a good question. I don't know the answer to that. Okay, so in this strip, the 1 lights up. That means that you're either a 1.1, a 1.2, or a 1.3. The 2 did not light up, the 3 did not light up, the 4 did not light up. That means you're not a 2, 3, or 4, but you are a 1.1, 1.2, or 1.3. Okay, here the control lights up, so that means the test's working. And then here we have a 1.1, that means you are a 1.1. You might be a 1.1, 1.1, or you could be a 1.1, 1.2, or you could also be a 1.1, 1.3. Here, we know you're not a 1.2, 1.3, because that didn't light up, and you're definitely not a 1.3, and this all but 1.3 means that you're a 1.1, so this person is a 1.1, 1.1. Now on with the countdown. I have no idea why I had Casey Kasem presenting that testimony, but the point I'm trying to make is it could be a little bit confusing. I mean, DNA analysis is brand new. It's a little bit, the analysts are trying to figure out how to present this in court. The jury members are trying to figure it out. You know, the probe way of DNA analysis is specific to an individual. This is not specific to an individual. So the conventional wisdom of today of, oh, everybody's DNA is different, that wasn't as well known or it wasn't part of a juror's, I guess, common knowledge. All right, so what happened? Well, before this caught on too much, the other thing that was going on in the mid-90s was they were using amplification techniques, to analyze short tandem repeats. And this is the method that caught on. The databases were built in the late 1990s, and it's still the technique that's used today, this short tandem repeats. Okay, so again, for those of you that are going to be attorneys, work in the court system, you have a bunch of cases that would have been investigated from the late 70s, early 80s, up through the mid 90s, where biological evidence would have been collected, but it was not analyzed using modern techniques. So if you have a defendant who's been in prison since 1992, they may say, hey, I want to reanalyze that evidence because I was convicted based on eyewitness testimony, and they can pull it out and do the modern analysis. Now, eventually, we're going to get far enough away from the 80s and mid 90s that we're not going to really have many cases that are still open. But especially within the last 10 years, 20 years, yeah, because that would take us back to what, 1997. So like within the past 10 or 15 years, reopening these cases and reanalyzing evidence has been a very advantageous thing to do for the forensic science community and the law enforcement community. Because when you go back and look at these old cases using DNA analysis, not only are you setting innocent people free and finding out perhaps who the real perpetrator was, it also sort of shines a light on how did these innocent people get put into prison anyway. Because we knew like DNA evidence didn't exist, so and we'll find out that something like bad forensic testimony on hair analysis, faulty eyewitness testimony, it could be leading interrogation of witnesses, well, not leading questioning of witnesses using mugshots in a certain way. So it really helped out the law enforcement community. Okay, now, any case that has been brought about in the late 90s to the current day, yeah, the techniques have gotten a little bit more sensitive, so maybe you can go back and get more information. But the thing is, if the analysis worked in 1998, there's nothing you can do today to get a different answer. Okay, you just get the same answer. It's only if something failed and you might want to go back and use one of the more sensitive techniques. But it's not as though there's been leaps and bounds in sensitivity of these tests. It's more or less remained very similar from, again, the late 90s, early 2000s. Okay, so the goal when a blood stain goes into the lab is to, number one, confirm it's blood, and then number two, produce a DNA profile for that blood stain. And I've mentioned this a little bit already, but the DNA profile is here in this piece of paper. This is just an example one that I use. Let's take a closer look at it. We'll see it's just one piece of paper. No, I thought it was going to be a bunch of A's, G's, C's, and T's. And no, it's not that. A lot of it is really not important stuff as far as a check mark on suspect and then nothing else. The important part is the DNA profile information. So you can see that's even a small part of this one-page piece of paper. And if we zoom in on this, we're going to find that we have the term locus, we have the term alleles, and we have a pair of numbers, a pair of numbers, a pair of numbers. Okay, and in fact, if you count these up, you have 15 pairs of numbers and then XY. All right, does somebody want to type in the chat box? What does XY tell us? All right, that tells us something about the individual. Okay, yeah, Sarah's correct. It tells us the biological sex of the person, and this person is male. All right, so these pairs of numbers here, and I'm trying to remember how much we got. I'm pretty sure it's mainly in this lecture and not in the previous lecture. But the locus is sort of the name of the location of DNA we're looking at. And these alleles, these pairs of numbers, you got one of these numbers from your father and one of them from your mother. So every time a human is made, from a forensic science perspective, at these different areas of DNA, you get these random numbers from your parents. Okay, they give you a number, and then your mother gives you another number, and then these are your numbers for life. Okay, if this was my profile, every time you analyze my blood, you'd get these pairs of numbers, these same 15 pairs of numbers. if you analyze my cheek cells, you'd get these numbers. If you cut off my leg and extracted cells from my bones, you'd get these pairs of numbers. Okay, get the general idea. The DNA profile is 13 pairs of numbers. Excuse me. So we want to know a little bit about where these pairs of numbers come from. Okay. The locations in the DNA, because the DNA is very big, and I think we've already talked about, the 3 billion base pairs and 99% similar and stuff like that. So we're looking at these locations in DNA that contain short tandem repeats. What does that mean? Well, we went over what short tandem repeat was at the end of the last lecture, but to repeat shortly and tandemly, it's like AGCT, so it's just 4 letters long, and it's repeated right next to each other. Okay, so if we zoom in on this location, everybody's going to have AGAT, 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 AGAT. It's just it will be repeated a different number of times in different individuals. And since we have 2 copies of the human genome at every location, that's why we get 1 set of repeats from our mother, 1 set from our father, and the result is that pair of numbers there. Okay, so don't worry, we're going to go over this a couple times. All right, so it's possible to do individual identification. We'll explain more about this later. These 13 pairs of numbers are what is uploaded into the FBI database. And I'm saying 13 pairs of numbers, that's what's uploaded into the database. The profile that we saw before had 2 extra loci analyzed, so it had 2 extra pairs of numbers. And then also the biological sex is uploaded. And the reason why individual identification is possible, to go back to our very first lecture, how many other people are going to have a 1315 and then a 1011 and then 7-8, or whatever those 13 pairs of numbers are, and the answer you can mathematically calculate it where nobody else is going to have it. Okay, and what we're talking about in the nuclear DNA in the chromosomes. Okay, we're going to talk more about mitochondrial DNA a little bit later. So again, this is what we showed in the previous lecture. Person 1, 2, and 3, they all have AGCT, but it's repeated a different number of times. Okay, but now we need to start thinking about what the actual representation of a person's going to be. And for person number 1, they're not just going to have AGCT, it's not just going to be repeated 6 times in their DNA, because in every single cell, they have a copy of DNA from their mother and a copy of DNA from their father. So this is actually a more accurate representation of what the person's going to see. Okay, we're going to analyze this location of DNA and find in person 1, they have it in the copy they got from their mother. AGCT is repeated 4 times. From their father, it's repeated 6 times. So if we were to describe this person's DNA at this location, we would say, oh, person number 1 is a 4 and a 6. Person number 2 is a 6 and a 7. Person number 3 is a 5 and a 5. Okay, so do you see that? Everybody is described by a pair of numbers. Okay, now we want to understand some terminology so we can start using the right terms to talk about these 13 pairs of numbers. Locus, or loci, plural, is the location of the STR in the genome, on the chromosomes. Okay, so you can think of locus as like a street address. Okay, if this is a map of your city, then the street address will take you to one location within this map of your city. Okay, so you may choose, okay, I'm going to choose a location on chromosome number 9. Okay, and there you go, right there. All right, the allele refers to the type of DNA. Now, alleles, if you remember your Mendel genetics from high school and your stupid P-pods and capital A, lowercase a, and all that stuff, okay, here the allele is not going to be a dominant and recessive gene. Here, the allele, the type of DNA, we're going to describe it by the number of repeats. Okay, so let's go over an actual example here. All right. Here, if we were describing a location on chromosome number 5, then we would again, go to that location. But since there's 2 copies of each chromosome, where one of these chromosomes you got from your mother, one of them you got from your father, you're going to have 2 alleles at each location. Okay, so one location, I mean, think of it as a street address. If I give you a street address, that's only one location on Earth, right? If I tell you to go to 123 Happy Lane in Homewood, Alabama, zip code 35222, I don't know what Homewood is. Okay, you type that into Google. There's one specific location. Okay, now imagine you went to that location and there were 2 houses. Okay, that would sort of be the same as in one location in your genome, you actually have 2 copies, 2 chromosomes. Okay, so you go to the location, there's 2 houses there, and for houses, the allele could be like color. Okay, so I give you a location, you go there and you say red, blue. Okay, I give you another location, you go there and you say green, green, 2 greenhouses. Okay, so again, location, you go there, there's 2 copies of what you're looking for. You're describing them in DNA as the number of repeats. Okay, each person has 2 copies. Is this lousy animation that I've done here? Yeah. Okay, each person has 2 copies of the chromosome, so each person has 2 alleles at each locus. All right, so let's look at some real names of loci, of locuses, and real examples of alleles. Okay, this is one of them. This is a locus, a location in the DNA, a short tandem repeat loci, where if you go to this location in my DNA, you're going to find AGAT repeated a certain number of times. If we go to this location, D5S818, in John Thomas's DNA, we're going to find AGAT repeated a certain number of times. If we go to this in Keith's DNA, we're going to find AGAT repeated a certain number of times. Does everybody get that? When we go to this location in Keith's DNA, we're not going to find just one chromosome there. We're not going to find just one allele there. We're going to find 2. Okay? And in a typical blood stain, like let's say Keith has bled all over the crime scene, in a blood stain, we're going to pick up, thousands and thousands of cells, which means we're going to have thousands and thousands of copies of this chromosome number 1, but they're all going to look the same when we zoom in on them. And we're going to have thousands and thousands of copies of chromosome number 2. That's all going to look the same when we zoom into them. So no matter how many cells we collect, no matter what body fluid we're collecting from Keith, when we do the analysis, we're always going to zoom in on that location, and it's always going to be a 7 and 9 from the day he's born to the day he dies, unless he has some weird robot transplant transfusion in the future. But that doesn't exist now, so we don't need to worry about it yet. Okay? So again, we want to convert a blood stain into pairs of numbers. Profile, which is pairs of numbers. So here, let's look for that D5. Where is it? Where is it? Oh, here it is. D5S818. This person, Mickey Spillane, is an 1113. All right. Maybe this is evidence from a crime scene. Okay. Hey, we can rule out Keith because he was a 7-9 at D5. Okay. Now, the problem is if we just looked at one location, all right, and Keith was a 7-9, it may turn out that Lori is also a 7-9. Okay. It's possible when you're just dealing with, again, and let's find our friend D5 here. Is that going to be a good example? Yeah, D5S818. Okay. You can be anywhere from a 7 to 15. So it's not as though you can have that repeated 2,300 times. Okay. So that's what? 7, 8, 9, 10, 11, 12, 13, 14, 15. That's 9 potential numbers that you could have. And you're going to pick 2 of them. You can see if you only have 9 options and you're going to pick 2 of them, you may coincidentally match somebody else walking around in society. And if you do the math, typically it's about 10% of people. You're going to be the same at that location. All right, now we're going to get into details later. But that's the reason why forensic scientists don't just look at one area, don't just look at one locus. They look at 13 loci. And this is the list of all 13 loci. What you'll notice is the names that they have are kind of funny. Sometimes they have a weird name like T-pox, which means the T is in pox is probably abbreviation for the gene that it happens to be in. All right, if it's listed, you'll notice a lot of them start with D and a number. D8S1179 means it's on chromosome number 8, the D8, and the 1179 refers to, I think, like the 1179th location described in the scientific literature or something like that. Okay, so they all have funny names, but again, we're going to think of these as the street addresses. They're on a bunch of different chromosomes. That's really for statistical reasons, which we won't get into in this class. You'll notice they all have 4 letters that are repeated, but we don't actually care about what the sequence is because we just care about what the repeat number is. And most of them are like from 6 to 15. Occasionally you can go up to 20 or 30, and D21 is a little bit weird because it goes out here from 24 to 38 repeats. But it doesn't matter what the repeat unit is. You're always going to get a pair of numbers. Okay, so if we analyze Dr. Linville's DNA at T-pox, I could be a 6'7", or a 6'12", or an 8'9", or a 13'13". You get the general idea? Okay, so blood goes in, 13 pairs of numbers come out on the report. What do? Do they just throw the blood into a machine with a big green button and hit the go, and then it starts, and it spits out that little piece of paper? No, you have to actually do some bench work, which means you take your pipettes, you dissolve your blood in a little bit of lysis buffer, bust open all the cells, and then you take it through a couple steps, and then another set of steps, and then another set of steps. Eventually, that sample goes into an instrument, and eventually, you can print out those 13 pairs of numbers. Okay. Now, I want you to think of all the stuff that's done in the lab as a 3-step process, okay? And we can see them here: extraction, amplification, separation. All right. Extraction is the first part, and again, we're not going to go into a lot of detail in JS110. This is something that we go into a lot more detail in JS352. But extraction is where you put your blood stain, or your semen stain, or bone dust, or a swab of the top of a cup, whatever you think has your cells on it. You put that in a tiny tube about the size of your thumb, and you add what's called lysis buffer. And it is a buffer with a little bit of soap in it and other chemicals that will break open all the cells. So essentially all of the DNA spills out of the cells into that liquid. Now if you did nothing else other than opening up the cells, there could be bacteria in there, it could degrade your DNA, there could be other things in the cell that could degrade the DNA. So you want to purify the DNA from all the other junk, both the junk that was inside the cells and the junk that may have been on the surface of the cup that you swabbed. Okay, the blood went on the floor. There may be a bunch of dirt and junk on the floor that you collected along with the blood. So you just want to purify the DNA. There are several methods of doing that. The method that we use in our lab, and that's automated in a lot of labs, is you pass it through this sort of silica gel membrane almost, although it's more of a disc. And the DNA sticks to the silica and all the junk washes through. And then you go back and you wash the DNA off the silica and catch it in a clean tube. Okay. But at the end of the extraction, what we have is you have a tube about the size of your thumb, and it just looks like it has water in it. Okay, the DNA at this point is not in solid form, it's dissolved in water. Okay, you take a little bit of that and squirt it into another tube and go on to the amplification step. All right, the amplification step is where and it's important to understand with the DNA extraction, you're extracting all of the DNA in the cell. So you're extracting all of the DNA from all 23 chromosomes. All right. If you have 10,000 cells in your blood sample that you started the extraction with, you now have 10,000 copies of each chromosome floating around and dissolved in that water. When you extract it, it usually breaks up into a bunch of different pieces, but that's not important. So what you need to do is, instead of 10,000 copies of that DNA, you actually need to only take about 350 copies, 500 copies or so, and squirt it into another tube. All right. And here you begin the amplification process. Now, the amplification process is not going to be where you amplify all of the DNA. Okay, so it's important to understand, if you get arrested for a crime and they take your sample and it goes to the forensic science laboratory, they're not amplifying all your DNA and are gonna, inject it into a clone frog and make a frog twin for you. Okay, that was a bad example. But they're not storing all of your DNA. They're not amplifying all of your DNA because they don't care about analyzing all of the DNA. Why? Because for 99.8% of the DNA locations, they're going to be exactly the same in all of us. So why even bother? They just want to focus on those areas of DNA that are going to be different among us. So they're only going to amplify those short tandem repeat areas. How do you do that? Hey, take 352. I mean, you put a little bit of your DNA, and these are the tubes. These are pipettes. So this is what you suck up a little bit of liquid with, and you inject a little bit of liquid with. All right. So you put a little bit of DNA. Hey, Bug, I'm teaching a class, so I can't play now. So you put a little bit of DNA in a tube. To be honest with you guys, I'd really rather go play. We've been playing school lately, and I've been learning a lot about ABCs and shapes and colors. It's fantastic. Anyhow, so you put a little bit of DNA in this tube, and you add what are called primers that will attach to the DNA only in the area that you want to amplify. And then you add some polymerase that'll replicate the DNA. But you stick this DNA, after you add your DNA and all of the components that will amplify and target only the areas that you want, you put it in something called a thermal cycler and you amplify it. How does the DNA amplify? Well, this box is literally something that heats up the tubes and then cools them down. It heats them up and cools them down. It's like a Planet Fitness commercial. I heat them up and cool them down, okay? Hey, that was hilarious 3 years ago. Anyhow, the heating and cooling, you can think of it as every time it heats and cools, it makes a copy. So if you start out with, let's say, 100 copies of the DNA, you heat it and cool it, now you have 200 copies. Then you heat it and cool it, now you have 400 copies. Heat, cool, 800. Do you see this double and double and double and double? Eventually it goes up and up and up and up. And at the end of about 30 cycles, you wind up with about 10 million copies of DNA. Okay, and this, you can see, is a lot easier to analyze and to visualize on the next instrument than if you were just chopping up those 1,000 copies and spreading it out on a gel. All right. Okay, so we amplified our DNA. We've amplified not one area of DNA, but 13 areas of DNA, 13 STR locations. Okay, now we need to measure those because we need to find out for each area that we amplified, is it 10 units long, 13 units long? Maybe it's 12 units long or 8 units. Okay, you guys get the general idea of how integers work, right? So in order to measure how long, how many repeats are there, you essentially race the DNA down a capillary and separate it, where the small fragments of DNA are going to go faster than the larger segments. Now, the book mentions this, and I have this in the lecture. Probably in the next version, the slab gel slide is going to go away because it's just becoming outdated. Everybody's moved to a capillary system, but the slab gel used to be done. It still can be done in some labs, and it's worth looking at the picture to sort of see how it's interpreted. What you would do is you would take your amplified DNA, you would put it in the big slab gel, okay, and then you'd race the DNA down the slab gel, and it would go in this, and it would, spread out, Okay, the smaller fragments of DNA would move faster than the larger fragments. So what you would end up getting is an image that looks something like this. Okay, the L's are the ladders. That's like the rulers, so we can ignore those. Okay, if we want to look to see what a profile from a person would look like, so let's say we got Jamie's DNA here, and Jamie is person number 1. Okay, well, at the location D16, okay, S1525 or whatever it is, okay, we can see that the ladder, if we read over here, is 5. It looks like 6 and 7 is skipped, and then this is 8, 9, 10, and we can see Jamie has a 10, 11, 12, 13, 14. Jamie has a 14. So at D16, Jamie would be, if he was person number 1, what the heck did I just say? I forget. A 10, 14. Pair of numbers. Got it? Okay. D7, let's see here. It's 6, 7, 8, 9, 10. Oh no, Jamie's a mutant, only has 1 band. No. Just, that means got a 10 from his mother and a 10 from his father. Okay, 10, 10. Again, a pair of numbers at D7. So that's how you would interpret this gel. And the software can sort of help read that out. But each of those bands are from a different area of DNA that you're then separating based on size. You can also do this in a capillary instrument. The capillary instrument, this is what we have and actually are still using in our lab right now. We got some updated instrumentation that's not yet online. But if you open up this door, open it up, okay, this is what it looks like. Here's a capillary. It starts in liquid here, and there's also an electrode going in there. And then the capillary runs, do you see it? Like here. And it goes through this detector window. So you're essentially racing your DNA through this capillary and seeing how long it takes to hit that detector window. Okay? The smaller the DNA, the faster it goes and the shorter time it takes to hit that detector window. When it hits the detector window, it winds up as a peak. So again, here's our location, and this person is an 1112 at D5S818. At D13, they're an 88. At D7, they're a 911. So at every location, you get a pair of numbers. Does that make sense? All right. Here, this shows sort of a zoomed out where you run that same ladder. That's like your ruler that you use to measure. And then this is from person 1. This is from person 2. And we can see that they're different people because they have different pairs of numbers. All right, so you go through the extraction, amplification, separation process for your blood stain, and you get a report that has 13 pairs of numbers. We're just looking at 4 of them for this slide in this exercise. If you have 3 suspects and you take suspects number 1's DNA, okay, collect it with a swab of their cheek cells, extract it, amplify it, separate it, you get 13 pairs of numbers for suspect 1. Suspect 2, collect their cells, extract, amplify, separate. Your report, 13 pairs of numbers for suspect number 2. And you can do it for, if you run out of suspects, you compare it to a database, right? Okay, mock case quiz coming up for reals next Monday. All right, how are these questions going to be phrased? They may be phrased, your blood stain is 7, 9, 10, 13, and 7, 15. And it does not match suspect number 1 at any of the loci. What is your conclusion? Well, our conclusion is, what is that? Faint? Is that shaded? I have no idea what's going on. Oh, no. Okay, I see. And suspect number. Do you like how I use the color contrast where the colors are almost the same exact colors? I'm sure in whatever PowerPoint I was doing this in, there was much more contrast here. Make sure your monitor is tilted in the right direction. It doesn't match suspect 1. It doesn't match suspect 2, but we can tell by the same faint purplish gray color that the blood stain matches suspect number 3. So what is our conclusion for the blood stain compared to suspect number 1? All right, well, I'm going to ask you guys. If I showed you blood stain in suspect number 1, what would your conclusion be? Did the blood stain definitely come from suspect 1, maybe came from suspect 1, or definitely did not come from suspect 1? What do you think your conclusion would be? Go ahead and throw it in the chat box while I take a drink of water. Definitely did not. Correct. Okay. Now, if I showed you the blood stain in suspect number 3, okay, matched at TPOXX, matched at CFS, matched at D5, matched at D8, if they match at all 13 loci, what are your conclusions for suspect number 3? Did it definitely come, maybe come, or definitely did not come from suspect 3? Definite match. Okay. You guys are correct, except that answer might not be correct on the quiz or exam. And the reason is, we're going to see in a couple slides, some states don't want you to say it definitely did come. And the reason is, there's a small chance that somebody is walking around out there that just happens to have the same profile. I mean, it could be an identical twin. If it's an identical twin, then if you know the suspect has an identical twin, then you don't want to say definite yes, right? But even in a normal circumstance, it might just so happen. And the preferred way is to have the forensic scientists, yeah, Sarah's right, they actually give statistics on the match. They say the blood stain matched the suspect, and this is the frequency of that profile. And it's like 1 in 3 trillion, so hopefully the jury goes, oh, well, there's only 7 billion people on Earth, so I guess that means it's not. And suspect number 3 was seen shouting at the dead person the night before, saying, I'm going to murder you tomorrow. So the jury puts all of that together. Okay. More on that later when we get to the end. Okay. So pay attention to that slide. Also for the quiz, stick around a little bit. Mitochondrial DNA. All right, this is something that forensic science labs could do, but don't necessarily do all the time. In Alabama, they do not have a mitochondrial DNA lab in the state of Alabama, but if they wanted to do this type of analysis, they would just package up the evidence and ship it to a lab or the FBI to do mitochondrial analysis. Okay, so those still would pay to get it done. But mitochondria is a little bit unique. So we're going to look about the advantages and disadvantages of using mitochondrial DNA. So again, short tandem repeats, the nuclear DNA, that's the standard. So with mitochondrial DNA, we just want to look at when would you do mitochondria in addition to STRs, or when would you do it instead? Okay. Why do mitochondria have their own DNA? It's where the oxidative phosphorylation goes on. It's the powerhouse that produces the energy. The theory is that mitochondria used to be single cells that got enveloped by a larger cell, and then they lost their function until they just sort of have this symbiotic relationship with the cell. But if you think about it, the mitochondria needs a whole bunch of proteins to complete its function. So as opposed to coding for the proteins here, making the proteins here in the ribosomes, and shipping the proteins to the mitochondria essentially just does everything itself right on the inside. Okay, it transcribes, it translates, it makes the proteins, and then it uses them. So it needs its own DNA. It's still a double helix, so don't get confused there. The difference is the mitochondria does not have chromosomes. So we're not going to look for STRs, and it's not going to be a pair of numbers. Okay, mitochondria is still, imagine the spiral continues, and the spiral continues, and it wraps around, and it forms a big circle. Okay, that's what this ring of DNA is here. Okay, this is what mitochondrial DNA looks like. It's similar to lower organisms, prokaryotic cells, but just imagine that this is sort of that double helix spiral just going all the way around. All right. So for mitochondrial DNA, we want to find areas in this mitochondrial DNA that is different among individuals. Now, the problem with mitochondrial DNA is that all of these areas are genes. And if we looked at all of these areas and compared my DNA, my mitochondrial DNA, to Jamie's, to John Thomas's, to Keith's, to Lori's, to Sarah, we would all have exactly the same, and what would it be? About 15,000 letters from here to here would be exactly the same. I think there's maybe like one letter that in some people are different. Okay. But in this D-loop region that doesn't code for the important proteins in our mitochondria, and the reason why it's conserved and we're not different is because if I had a unique COX-5, okay, or what is that, COX-3, okay, cytochrome oxidase 3, if my cytochrome oxidase 3 was a little bit different than yours, that wouldn't be a good thing. It means it wouldn't function and I would be dead. All right, so that's why we need to be the same there. But in this D-loop region, it doesn't code for proteins, and it's free to be a little bit different among us. But again, there's not that much difference. I mean, we're talking 900 bases is the D-loop, only a 1.7% difference. So if I compare my mitochondrial DNA to Sarah's mitochondrial DNA, we might have like 18 base pairs that are different or something like that. Okay, so it's not going to be a lot. And it could be significantly less than that. All right, so nuclear is the double helix, mitochondrial is the double helix. Nuclear is 46 chromosomes, mitochondrial is just that one ring. Okay, now here, the copy number. This is what's important and gives mitochondrial DNA an advantage. In nuclear DNA, you just have one copy per cell. Well, that's actually not quite wrong. It's two copies, one copy from your mother and one copy from your father, but your entire nuclear DNA, there's just one of it. One set is maybe a better way of putting it, one set per cell. In mitochondrial DNA, that ring, there could be like 10 rings in a mitochondria. And in each cell, there could be 10 mitochondria in a cell. So in other words, if you have a sample, okay, an evidence sample, let's say it's like a hair or something, and you only have DNA from like 3 cells in that sample. DNA from 3 cells is not enough for the amplification to work. You're going to try to amplify the nuclear DNA, and it's going to fail. Okay, but in only 3 cells, if you have 10 copies in each mitochondria, 10 mitochondria in each cell, that's 100 mitochondrial DNAs in each cell. That's 300 copies of mitochondrial DNA. You may be able to amplify mitochondrial DNA in that situation. Okay, so one reason to use mitochondrial DNA is in older degraded samples. If you try to amplify the STRs and it fails, then maybe we can try to amplify mitochondrial DNA. Okay. Now that 900 base pair region that I talked about, there's no STRs in that. Okay, so there's no pairs of numbers, there's no one number. Mitochondrial DNA analysis is more or less what you would think of and what we went over in the last lecture as far as the bases being different. Okay, so you would actually, what's my mitochondrial DNA profile? You would write out 900 letters. Okay, and then you would get mitochondrial DNA from the evidence and you would write out 900 letters. And then you would compare my 900 letters to those 900 letters. Now again, no matter what the human is, probably about, 880 of those letters are going to be exactly the same. But there could be enough differences that my mitochondrial DNA is going to be different from Sarah's and different from Lori's and different from Keith's. How do we get that sequence? The extraction is exactly the same. Whatever our sample is, be it a hair, be it an old blood stain, be it a bone, an old degraded bone. Okay, we grind up the cells, put it in a tube with buffer, separate the DNA from the junk. Amplification is very similar to the same. Actually, whoops. Can you tell? I just copied and pasted this slide. Do this. No, we're not going to amplify STR regions. That's wrong. All right. we're going to amplify that 900 base pair area of DNA. So we're actually going to just put in one primer set that will amplify that 900 base pair area. And then we put it through a somewhat complicated process called sequencing, but it's still where we feed it into an instrument and it spits a sequence out. Okay, so don't worry too much about that. Then we compare the sequences. We have a sequence from our hair, and this would actually be 900 letters long, not just enough to fit on the slide. A sequence from our victim, if we're trying to link this, let's say that we found this hair in our suspect's car, the trunk of the suspect's car, and we think it matches the victim. All right, so we try STR analysis on it, but it fails. If we compare it and the sequences match, then, okay, now here, if the sequences match, what is your conclusion? Did it definitely come from the victim? Did it maybe come from the victim, or did it definitely not come from the victim? What do you guys think? And the answer is sort of up there. The answer is maybe. And the reason it's may have, and the reason it's maybe, is, well, are there any other people that have that same mitochondrial DNA profile? Yeah, there are, because we're dealing with only 900 letters, only 18 that are potentially different. And they're in the same location. So it's not just like 18 random of those letters. It's sort of like 18 within there usually have the differences. So it's possible that a stranger may have the same sequence as you. Okay, but what's guaranteed is that, and it doesn't say it on this slide, maybe this is in the next one. What's guaranteed is that somebody in your family has the same exact mitochondrial sequence, because mitochondrial DNA is inherited from the mother. All right, I'm going to wait to explain this in the next couple slides because I think it's coming here. All right. What if it doesn't match? What if one letter is different? Okay, here if we have a difference, we need to ask ourselves, well, can that trait change over time? Can DNA change over time? Is it possible that the DNA in the hair from the victim has changed by the time we find the victim's body and analyze that? Okay, DNA does not change over time, right? Okay, so what is the conclusion here? The conclusion, as it says up there, you can say definitely no. Just like if the STRs don't match, you can say definitely no. Sometimes people don't want to, if I phrase this as 899 of the letters match, but only one letter does not match, what is your conclusion? The conclusion is still definitely no. Okay? Don't let those other 899 influence you. Okay? It has to be exactly the same for it to be a match. All right? So here, this sort of shows, okay, we got mitochondrial DNA from a cigar, maybe just a little bit of saliva on it. DNA analysis, STR analysis fails. And here we have a person of interest. Okay, 1, 2, 3, and 4. All right, what do we do? Well, we can rule out. Did the cigar belong to person of interest? Okay, let's not say belong, but was person 1's DNA on the cigar? Yes or no? Definitely no, because one of them is different. Was person's 3 DNA on the cigar? Definitely no. But can we say it definitely matched person 2? No. It may have matched person 2 or person 4, or it could be another person, their cousin or a random stranger that happens to have the same sequence. Okay? So that's the big difference between mitochondrial DNA and nuclear DNA. All right, now the reason for this, because I thought it was going to be in the next slide, but I don't think it is. The reason why you may be the same as other people is that mitochondrial DNA is inherited maternally. Here, wait, let me look at the statistics here. Okay. Hey, the good news is we're almost done. The bad news is Linville decided to go back and explain mitochondrial DNA. All right, so what did I do? I lost my little thing here. All right, again, this is going to be a graphic picture here. And there we go. Okay, sperm cell, here it comes towards the egg cell. All right, one copy of your nuclear DNA is packed in that sperm cell. One copy of your nuclear DNA is packed in that egg cell. When they come together, boom, you get 2 copies of DNA, 2 of each chromosome, like we talked about before, in the nucleus. What about the mitochondria? Check this out. The mitochondria are in the egg, but they are not in the sperm. Okay? Your mitochondrial DNA, whether you're male or female, matches your biological mother. Okay? So my mitochondrial DNA is going to be exactly the same as my mother's mitochondrial DNA. It's going to be the same as my sister's, the same as my sister's children. My mitochondrial DNA is going to be the same as my brother, but not my brother's children, because that's going to be his wife, because he can't pass it down. My children don't have my mitochondrial DNA, despite my efforts to genetically manipulate it and somehow no, I didn't do anything of the sort yet. My early experiments on mice failed miserably, leading to that one accident on that fateful night. And I have the nibbles on my toes. What am I talking about? I have no idea. Let's wrap this up. Yeah, I showed you the last slide, right? Okay. So all your maternal relatives are going to be exactly the same. Okay. Or, again, it could be a stranger that's the same profile. All right, let's go back to nuclear DNA. Okay, notice that this is a trick, because I'm saying that if this is a mock case question, you cannot say the DNA in the bloodstain is definitely John Doe's DNA. Some states won't let you do that. What they prefer you say is that the frequency of this, it's number one, the profile from the evidence match the profile from the defendant or the suspect, and the frequency of this profile is 1 in 300 trillion. And it's not a number that they make up. It's a number that's actually calculated. And to do that calculation, I mean, we could get into the statistics of rolling dice or flipping a coin and getting heads 50 times in a row. But what you do is you figure out the percentage for each loci. Okay, and like I said, this is always going to be like around 10%. So we just have some examples here. All right, so you calculate it. You look at the frequency of the alleles. If it's a heterozygote, you multiply by 2. Don't worry too much about this slide. I used to include more of this in questions, but I don't have it anymore. But what I do want you guys to understand is, okay, at D5, my DNA is going to be similar to 18% of the population. And at D8, my profile is going to be similar to 13% of the population. And at D18, oh, my profile is a little bit unique. It's going to be similar to just 0.6% of the population. Okay? So there's like 18 out of 100 people matching you, 13 out of 100 people matching you, 6 out of 1,000 people matching you here. That's still not unique, right? But if you look at all 3 of these, and you ask yourselves, how many people match me at D5 and D8 and D18? Then what you do is you multiply these percentages together. All of a sudden, your frequency is 0.0001, or what is that? 1 in 10,000. Okay? 1 in 10, 1 in 100, 1 in 1,000, 1 in 10,000. So you can imagine the more loci you use, this decimal point goes over and over and over and over and over, and pretty soon your frequency has 13 zeros or more in there, okay? And that's where you get that 1 in a trillion, 1 in 300 trillion, 1 in 4.5 quadrillion. And oftentimes when you read newspaper articles where they're talking about forensic scientists' testimony on DNA analysis, they'll put that number in and they'll go, oh, look at this, he's making up numbers. No, quadrillion is a real number. Okay. So there are tables forensic scientists use that list these allele frequencies, you know, how many times you find 10 in a population or 11 in a population for given genotypes. And they actually sit down and they can calculate those numbers. All right, so we'll close with this. On a mock case quiz, if I'm giving you STR information, okay, and I say you receive 13 numbers from the evidence, and 13 pairs of numbers from the suspect, and they all match, what is your conclusion? Definite yes, maybe, definitely no, or you would tell the jury they match and calculate the frequency of the genotype. Okay. Yeah, be careful because it's not really a maybe, because maybe implies that it might be somebody else, and you don't really want to say that because you wanted the jury to come to the conclusion that, oh, it matches this guy and nobody else. So it's sort of where you're not saying definite yes, but you're giving them the impression that it's a little bit more than a maybe. Okay. So again, this is one of those where the phrasing, you're going to choose that answer with the weird phrasing in the question. Okay, now here's the one that everybody always misses. All right, you guys want the inside track here? You calculate the profile from the evidence, and you calculate and you produce 13 pairs of numbers. You process the suspect and get 13 pairs of numbers. When you compare those, 8 pairs of the numbers match, but 5 pairs of the numbers do not match. What is your conclusion? Definitely yes came from the suspect. Definitely no. It might have come from the suspect. Or you would do some frequency crap. What do you guys think? Because for that situation, everybody always wants to see, oh, 8 of them match. Maybe it came from the person. But remember what we said before. If just one of the loci that you look at does not match, DNA cannot change over time. I can't be an 8-9 when I'm bleeding at a crime scene, and then later when you pick me up, my DNA is magically changed to a 10-11. Okay, so remember, you guys, that's a definite no. Mitochondrial DNA, you compare them. If there's just one letter that doesn't match, it's a definite no. And for mitochondrial DNA, if everything matches, that's a maybe because there are other people walking around with the same profile. Here, and I can tell you guys, for the database, there's like over 1 million people in the database. I'm sure they've reached over 2 million by this point. They've never had 2 random people match at all 13 of those loci. Okay, the statistics show that it won't happen. It would be extremely rare, and they haven't seen it yet. And they've been comparing, profiles for 15 years. It'd be a big story if it did, but other than identical twins, they're not going to match. Okay, that's it. We ran DNA part 2 is always a little bit long, so I'm going to go ahead and stop there for tonight. Hopefully this will help you out with the mock case quiz coming up next week. I'll leave this running a little bit more if you guys have any quick questions for me. I will answer on the recording. Actually, nobody leave until I call your name as well. And I'm going to update my attendance because I think a couple people snuck in that I did not record. Let's see here. Let me start at the top. All right, so Isabel, I have you down. Jamila, I have you down, and after I call your name, you can leave the room. All right, Jamie, I have you down. John Thomas, I have you down. Keith, I have you down. Lori, let me write you down. All right. And Sarah, I got you down. All right, I'm going to go ahead and stop the recording now.